Okay, so I think we will get started, and then as people come in, we will just let them in. So my name is Ellie. I am the president of the Pdx Ducks, and I just want to thank everyone for coming to another science night. I'm so happy you could make it for our Halloween themed event. The Pdx Ducks are a dedicated board that strives to engage, support, and maintain connections with local ducks. So make sure you're checking your emails and following us on social media to keep up to date with everything that we are currently doing. So we will be taking questions at the end. So if you have a question, just say that you have a question in the chat, and then Sandy will call on you at the end of the presentation for you to ask your question. And now we will be handing out 2 gift cards as well. So please send us a private message with your email. You can just message me on the Zoom function when we call the winners. So now to introduce Sandy Rice, who is our Science Night Chair.
Thank you, Ellie. And I appreciate those of you that did dress up, so I'm not by myself in my little worm outfit as the scientist that hasn't had any experiments go well. So we are very thrilled that we were able to snag our Provost Patrick Phillips to join us and tell us about his current work at the university. UO Provost and Senior Vice President Patrick Phillips, a member of the Institute of Ecology and Evolution and a member of the NIH Center for Excellence in Systems Biology, is a nationally recognized expert in ecology and evolution. His research focuses on the biology of aging, genomics, and the genetics of complex traits, and he is working toward discovering how to increase people's health spans and decrease the impacts of environmental stress. Once again, we are thrilled to have him here, and I will turn it over to him.
Great, thank you so much, and thanks to everybody who's joining us. Let me just make sure I get my PowerPoint up. Almost there. There we go. Yes, those of you who are looking through, if you see Levi Moran on there, he's my former PhD student. He's now an associate professor at Emory University. I'm very proud of his accomplishments. He's a great duck. And I did not know he was going to be on this call. And he helped establish a tradition. Now, this is the 3rd time where he dresses up as me for Halloween. So luckily I can't see Levi's face right at the moment because it was his little picture was freaking me out a little bit. So I will ignore Levi and move on. And I do appreciate everybody, especially those of you who dressed up, and especially everybody who came. And I am provost, so I also want to thank you for all of your engagement and involvement in the University of Oregon. It's a great university, and I'm proud of it. I've been here for 20 years. I am a member of the biology department, and I've been provost for about 1.5 years now. So in that role, I get to see all of the university, and it's really an exciting place. So what I wanted to do today, I was invited to talk about the work that we've been doing in the field of aging. And this picture is of the Egyptian god Anubis, who is the god of death in Egypt. And here is a picture of Anubis weighing the heart. This is part of the reason why your heart is removed. Well, not you personally, but during the mummification process, so that Anubis can weigh your heart versus the feather of truth. So the feather is quite light, and you want to make sure that your heart is light and because of your good deeds when you die. And so there's some parable there going into an election week that we are doing, but I will not talk about that. I'm going to talk about my own work, but I would be remiss if I didn't talk about actually perhaps the most exciting science thing going on at the University of Oregon right now, even as we speak. And that is, in this age of COVID, why am I not visiting with you in Portland right now, is because we're all being safe. So we have set up an entire testing facility at the University of Oregon. And so a group of faculty and staff have set this up just over the last few months. It's a CLIA certified medical testing lab. It's something that we've never done at the university before. And so it has two parts. One is the people who are out collecting the samples. And then we have a whole genomic testing facility. And down in the right-hand corner, you see Doug Turnbull, who's the head of our genomics facility. And there's the lead geneticist on campus, Mike Schill, who has been learning a lot about public health over the last 6 months. So President Schill has been a great leader for us during this COVID time. And they're looking at a robot. And so when COVID hit, Lane County only had the capacity to test about 100 people a day. And that was actually below the threshold needed to actually even open up from quarantine. So the university stepped forward, we bought some equipment and we partnered with Mackenzie Willamette, and we immediately raised that to 500 tests a day. So we quintupled the amount of capacity. And then we set up our own lab. And so the combined capacity right now, as I speak to you, between Mackenzie Willamette and us is about 1,200 tests a day. And so, for instance, on move-in for all of our students into the dorms, we tested every single student in the dorm twice on move-in so that they could be safe. And fortunately, our students were extremely well behaved, and the positive rate was very, very low. Because we had done all this great work, the state of Oregon came to us, and we've been partnering with them. Excuse me, I'm going to have a sip. This is the robot in action. So this is what it takes to do thousands and thousands of tests, is you have to really get a lot of work done and process a lot of samples. So Oregon just recently gave us a contract for $4.3 million to buy even more equipment so that we can now ramp up to as many 10,000 tests a day. So that will be a great service to Lane County, and I expect the state as a whole. And so that's an ongoing partnership. So this is a great illustration of why research university, and especially one that's very strong in the sciences, is so critical to a state like Oregon, because you never know what's going to come. And we were able to very rapidly pivot and serve the entire state with this effort. So now let me transition to my own work, which is involves a lot of different things, but I'm going to talk to you about our aging work. And the first thing that I want to make clear is that I help direct the lab. I'm a busy person now, especially as provost, but I have many people in my lab, and this is why we do it. So I have a lot of students, undergrads, graduate students, postdoctoral fellows, and also a lot of research associates that help get all the work done. So I'm not going to take credit for all of their work, but instead I'm here to celebrate all the hard work that they have helped me accomplish. So I want you to just take a second and take a deep breath and think about yourself. And usually when we do that, we think of who we are as a person. And during these times, we have a lot of alone time to think about that. But I actually want you to think about what you are as an organism and how you are made. So this is an early frog embryo coming into being. It's essentially the same process that you did some number of years ago. And we are remarkable, incredibly remarkable. So we're made up of about 50 trillion cells. That's a lot. And if you think about it, every cell has its own copy of DNA. And no cell, there's no master that tells what a cell to do. Every cell has to understand where it is and how to function by talking to other cells or remembering where it is in the body. And so that is a remarkable thing, our complex nature. So how does a cell on your nose know that it's on your nose and not on your rear end? So it would be embarrassing if it was the other way around. And so it's really important, but is also then the basis of really all complex life, but also the underpinnings of what it is to get old. So this communication, this holistic way that we exist, what tends to happen through aging, and this will be a theme that I'll point on, is that we start to lose our individual coherence as an organism as we get older, and that's one of the challenges. So we recognize that aging is a thing that happens. This is my favorite scientist, Charles Darwin. I kind of dressed up, I guess, like the middle-aged Darwin, but with more hair is one way of looking at it. Hopefully I won't quite look like the old forlorn Darwin later in life. But we're very used to this trajectory, right? We start off young, we look a certain way, and then we just assume that we're going to get older. And then, of course, eventually we're all going to die. And so that seems to be a reality of our lives. But remember, as a biologist, and we're all biologists now tonight, we should ask why? What happens? What is aging? That seems like we take it for granted, like everything that we do as organisms. But what does it mean to get old? That's really a fundamental question. So this is Kane Tanaka. This is the oldest person currently living. She's almost 118 years old. She's Japanese. As you know, there are many people over 100 in Japan. But I want you to think about Kane for a second. So every single person that was born before she was born is now died. Since she was born, 15 billion people have been born in this world. So if you think about it, she is a fulcrum in time in which everybody preceding her is gone and everybody who is here now has been born since she was. And in many ways, that is each of our lives. Every life, every birth, every death provides a fulcrum or a nexus in the way that we think about our personal histories and the way that the world is actually structured. And so it's not surprising that sort of birth and death are the central themes of almost everything that we do in our culture. And of course, the in-between parts is the work that we do and the fun that we have. So, so it's really worth thinking about what that means. And someone like Kane is obviously exceptional to be 118 years old, but for sure throughout human history, it's very exceptional. So if we look at most of the history of humans, there were almost nobody that would live that old. As a matter of fact, we probably died pretty young. So if you look at the causes of death in countries such as sub-Saharan Africa, we can see that what happens is we have things like lower respiratory infection, that is pneumonia, that would mostly be in children. There are some diseases, especially related to heart disease, that are still prevalent. But then we have diseases like AIDS, tuberculosis, infection, malaria, and then again, other early childhood problems such as low birth weight. So what we see is that in those countries, the major causes of death are either infection or something due to problems early in life because children are particularly susceptible. And that's probably matches what has been the case for our vast majority of human history. If you look at modern societies, especially in Europe and North America, where there's higher income, especially over the last 200 years, we see that the profile of what people die of has changed dramatically. So heart disease, which was present in Africa, of course, but now is the #1 cause by a lot, and other cardiovascular diseases such as stroke. But now we see some other things, cancer. We still have lower respiratory infections, but this is mostly killing the elderly, not children now. And then we have a new kind of disease, Alzheimer's or other dementias, cancer, diabetes, cancer. So the profile of what kills people is completely changed in the last, especially 100 years. So we could say that these are diseases of modern society, and some people might say that, for instance, cancers, but that is not really an accurate depiction. What is really the case is that these are byproducts of being in a modern society in which people live longer and longer. And so these are the diseases that start becoming more relevant. So after you take care of infectious disease, then there are many other things, and they're mostly diseases of age. So when we look at the distribution of lifespans across the world, and this is kind of small depending on what kind of screen you're on, hopefully not your phone, things that are in green are countries where there's long life. So for instance, in the dark green areas, those are average life expectancies over 80 years old. Anything over green that's colored green, say light green and above, is 70-year-olds or more. The red colors are in the 50s, and the dark red is under 45 years old. So we can see that there's a lot of difference in just longevity across the world. And those are not due to biology. Those are due to socioeconomic factors that then layer on top of really the prevalence of infectious disease and malnutrition. But again, historically, we were more like the red countries than the green countries. So this is a graph that shows the change in life expectancy over the last 150 years or so in European countries, and USA comes in the end here, so it's a pseudo-European country. So what we see is that in the 1840s, the average life expectancy was about 40 years old, maybe 45 years old, okay? And so that's what we would expect. And now, and this actually cuts off at 2010, life expectancy in a lot of European countries exceeds 80 years old and into 85, 86 in some countries. So it's really an extraordinary increase. That's the first thing I want you to take away. A doubling of lifespan in the last 150 years or so. So that's really extraordinary, again, from us as biological organisms. And then the other feature of this graph, I think you will note right away, is that this is a linear increase and it's not stopping. So our longevity just keeps going up and up and up. And I think that you have probably all experienced that. So what happens as we get older is that the prevalence of some diseases also start increasing in frequency. So here are three diseases that are essentially related to older age. The first one is cardiovascular disease, which some young people get in 16 to 24 even. There's maybe a few percent. But as you get up to 75 and above, and the blue is in men, and the pink or the oranger color is in women, you get 40%. Dementia is, of course, the most striking, where essentially dementia is unknown in people earlier than 60 years old, whereas you get older and older, when you're in your 90s, the prevalence in women can go up to 50%. When we look at cancer, that is also, obviously there are childhood cancers, but the prevalence of cancer goes up exponentially late in life. So one way of thinking about it is that we have whole research programs that are built around addressing each of these diseases. But a different way of thinking about the problem that a lot of biologists are starting to say is, what if we think about aging as the primary factor? What about aging is driving disease? So if you could treat aging per se, that you might be able to get at that. But that raises this really fundamental question, and that is, what is aging? What do I mean by that? And so we think, well, it's getting old. But what is getting old? And this is the way that biologists, especially demographers, think about it. So I'm going to plot out what the chance of living to be 100 years old is in the United States. So if we use what we could call the bus model of longevity, that is, we are all living, and then we just have a random chance of dying because we get hit by a bus. There's nothing else about biology. It's just random chance. If we all start with everybody being alive at age 0, what would it look like if they were 100 years old? And so the constant mortality model looks like this, that essentially there's a decreasing chance that you'll be alive because there's just too many buses in the world. But that is not at all what we see, right? So that is not the way that human longevity looks. Instead, it looks like this, where we have most people are alive early in life, and then especially in the United States, you're going to live for a while, and then around 60 years old, something changes. And then the rate of mortality dramatically increases late in life. So I want you to think about what that means. Sorry, my screen is not happy about.
Just about to say, we're not getting a full slide anymore.
Yeah, let me just end the show and then reshare. The graphics seem to be freaking out a little bit. This is life in the modern era here. Now
Gives us a chance to see everybody.
Yeah, that's good. All right, let me try it again. Okay, first that, on that. Sorry, I have two screens. So if you've ever done that before, you know, just finding your mouse is a major activity. Okay, here we go. Are we back on it?
Yes.
Okay, good.
We can see it.
All right, so hopefully you can see these two curves for men and women. And we know that men don't live quite as long as women, but overall the shape of their curves is the same. So what I did with this, this next flatter line is imagine that we took the probability of a man living to be 60 years old and then just froze them in time and said, okay, let's just hold that probability and extend it out in time. How long would they live? So it's 250 years. So if we could just keep the average US man to be at the same physiological state that they are at 60, they would live to be 250 years old. So what that tells you immediately is that this decline is changing very rapidly above 60 years old. So I wanted to do this thought experiment instead. So imagine that we stopped at 30 years old and then said, okay, we could capture your body and your physiological state at 30 years old. How long would you live based on that? 1,700 years, okay? So that's pretty remarkable. So we could say, well, it's crazy that people could live long or live like elves or whatever you want to say. But in fact, there are people on this planet right now who have the physiology to live to be 1,700 years old. They're called 30-year-olds. So something is changing in our biology that is driving this aging process. So the question is aging inevitable? Is it biological? Is it programmed? What we do know is that something is driving this, and that we have a word for that. We call it senescence. So there's an accelerated rate of mortality. That's what aging is. It's not just getting old, it's getting old faster. Okay, so that's what's driving the question. And we do know biologically that other forms of aging are possible. So this is a Bristlecone Pine. You may know about this one. The oldest living organism, depending on how you want to count it, but it's a single independent organism, was a Bristlecone Pine. It lived 4,800 years old. That's a long time, even longer than our theoretical 30-year-old. And how did we find out how old it was? We counted its ring. So it turned out that what happened is that a graduate student from North Carolina, thank God not OSU, went out in California and took a borer in the 60s into this tree to see how old it was. Unfortunately, the boring instrument got stuck. So they cut down the tree to get out their borer. And then they discovered they had just killed the oldest living organism on the planet. That was generally considered a bad idea. But anyways, that's how we know how old the trees are. So we know that the tree can live that long. But then we have other kinds of organisms. So a meadow vole, just like any kind of rodent, doesn't live a particularly long time, maybe 1 to 2 months. But not that distant relative, the truly Halloween-ready naked mole rat lives to be 20 years old. So many, many orders of magnitude. So they have essentially the same biology. They're both rodents. They kind of look the same, plus or minus hair. But something is dramatically different about the biology of a naked mole rat that allows it to live a lot longer. And believe me, there are biologists who are looking into this. So what is going on? Can we actually understand that? And if we could we actually change ourselves from a meadow vole into a naked mole rat without the egregious consequences? So what essentially is going on with us is that we age because we are starting to fight against ourselves as coherent multicellular animals. And so this is one of my favorite paintings. It's by Peter Bruegel. It's called The Fall of the Rebel Angels. And so this is essentially describes the story of when everybody was in heaven and everything was great. And then, you know, something bad happened, and then we had to drive out some of the angels, and they became essentially devils, right? So that's that story. So that's essentially what's happening within us in almost all these diseases, is that something's going wrong in our bodies, And frequently what happens is that our bodies are fighting against each other. And of course, you all know that the classic example of that is cancer. And there are many other diseases that we know that when you break some feature of the organism, you can age quickly. So here's one example called Werner Syndrome. It's involved in a DNA repair system. And so when you have a mutation in this particular protein, you don't undergo proper DNA repair. And this is a woman who showed signs of very rapid aging because essentially her body could not regenerate itself coherently. The most extreme cases are progeria, and these are 7 and 8-year-old children at the bottom. So this is a nuclear laminin that basically their cells can't keep their coherence together, and they have this very rapid aging phenotype. So this is kind of the state of biology for a very long time, that we knew that there was lots of ways to make things go wrong. The revolution that occurred about 30 years ago is that somebody figured out that organisms can make things go right, and that changed the way that the field progresses. So this is this little worm, and that's I really appreciate the support from the alumni groups here that they came fully decked out as worms. And so this is this little nematode worm, C. elegans, that's now one of the most studied organisms on the planet. Why do it, and why study it in the context of aging? Well, it's because of this discovery that was made. So here's how a worm lives. It's just very similar to those kinds of curves that I was showing you about humans, except that worms live much less long, maybe only about 3 weeks instead of decades. And so that actually turns out to be a great experimental feature, because if you think about doing an experiment, even a mouse will live several years in the lab. So it takes a very long time to do aging experiments. So that's part of the reason why worms are popular. But here's the real reason. Someone discovered a mutation called DAF2 that allows the worms to live twice as long. So this was a genetic change. Instead of making things age more quickly, it made them age more slowly. So that suggested that there was something in our genes that could be tapped to make us live longer. And so that started an entire revolution in the field. And I think I will skip ahead just a little bit. I was going to tell you how this works, but essentially what happens is the DAF2 gene is involved. It's actually an insulin receptor. And so when it's active, it tells the worm that there's lots of food around and you should just go about your life. But when it's disrupted, then it activates a stress response because it tells the worm that it's in a starvation condition. And then that upregulates, as we say in genetics terms, the stress response genes. So what the worm told us is that if you turn on these stress response genes when they're not necessarily needed, that all of a sudden you can make the worm live longer. Makes sense, right? You're turning on a system to protect you from stress, and so it protects you from stress longer. So why wouldn't worms always do this? It's because it costs energy. And the currency of biology is how quickly you can reproduce and how many offspring you can produce. So you don't necessarily want to invest a lot of energy in keeping your body alive if it comes at the cost of your own reproduction. Humans don't have that issue because we live, at least in Western societies, in areas of abundant energy. We can just open another pack of Fritos if we want, right? So if we could turn on systems, even if they took a lot of internal energy, then that would make us live longer, then that would be fine. At the same time these discoveries were being made, another study was started in mice to see if we could figure out how to make mice live longer. The first thing they discovered, this is not the way to make mice live longer. So what they started doing is giving mice different compounds and drugs to see if it would extend their longevity. And they did find some that are really interesting, and those are actually starting to move into human clinical trials. But one of the things that they discovered at the same time is that when they used a drug on one mouse, all the mice are essentially genetically identical. And then they moved to a different genetic background. Sometimes it didn't work, okay? And it turns out this is a general feature of medicine. So this is a diagram that shows the 10 most, I think these are the most profitable drugs, that is with the highest sales, not necessarily the most used or the most important. So you see Abilify, Nexium, Humira, Crestor, Cymbalta, Enbrel. You also see these in TV commercials. And I have little doubt that some of you take some of these for sure. So what this graph shows is how effective they are in the human population. So basically, the little blue person represents it works, and the red people show the percentage of the population that it has no effect on, that it doesn't help those people. So I think you can see the general pattern is it's great that these drugs work for some people, but even the most effective drugs only help about 1/4 of the people who are treated with them. So something is different among people that makes drugs not work for particular diseases or syndromes. And so that's a problem. And this is a problem in medicine. It's actually a major feature of the way that medicine is progressing now. So the NIH, who funded the mouse study, said, oh, we got to figure this out for these aging drugs. And turns out that mouse experiments are very expensive and time consuming. And so they turned to worms because it was a model system. And so my lab is one of 3 labs across the country that is part of this big NIH study to try and find compounds to make worms live longer. And it's the kind of thing that when you hear that in congressional testimony, people start making fun of you. But of course, what we're trying to do is find compounds that make humans live longer, healthier lives. And this is the means to do that. So we have different strains of worms collected from all over the world. They're genetically different. We actually have multiple different species of them. And then we test them with many different drugs. So I'll just tell you just a couple of quick stories of what we found. Here's one drug called thioflavin T. It's actually a dye that binds proteins. And what this graph shows, and I won't have a lot of graphs because they can get boring, is basically 6 different genetic strains of worms, actually 3 from 2 different species each. And the little dotted line across the middle there shows what the control is, that is if you don't treat them. And then this is the amount of increase. The little different colors are different experiments in the different labs. And that's another big feature of our study, is that we actually look at how repeatable the work is in addition to whether or not we see an effect. So overall, you see that the drug actually makes everybody live longer, which that's great, but it varies a lot based on genetic background. So sometimes it can close to, well, about 75% increase in your life. Sometimes it's pretty wimpy, just a few percent increase. So that's interesting. So that's cool. And so we do these experiments by actually putting the drugs on these little plates. And then the technicians in my lab, actually, this is the crappiest part of the whole study, is they have to find out if the worms are dead. Seems like it'd be easy, but the worms are, as they get old, they're not so active. And so you have to poke them and say, are you dead yet? And they say, no, not dead. So you have to basically test them. So we have a new technology in which we put the worms on flatbed scanners, and then the worms get scanned every 15 minutes. And then we just ask if they've moved in that 15 minutes. And if they stop moving for a while, then we say that they are no longer alive. So we did that with this drug. And instead of extending life, it killed every single worm within 1 day. Like, that doesn't seem good. So it turns out that the drug actually makes the worms photosensitive. So what we have is a drug that makes worms live longer, but they can't go out in the sun. Okay, so they're somewhat immortal, can't go out in the sun. Turns out we already know something like that. They're called vampires, right? So they live a long time, but they can't go out in the sun. Now, this is actually a dye, and so it makes the worms a little shiny. And so maybe we need this more modern kind of vampire, the sparkly vampire version for this. So it turns out that when we put sunscreen on the worms, that is, we put a UV filter on the light, then the drug turns to working again. So, but that's the kind of complexities that we have. So I'm not recommending that you take that one right now. Another drug that's gotten a lot of attention over the years is this thing called resveratrol. And resveratrol is interesting because it's a component of red wine. And so we tested it and we found that it has some mild benefits in one species, but not in the other at all. And again, so should you go out and just start drinking wine like crazy, maybe on a night like tonight? Maybe you are doing that right now. Sure. But you have to drink gallons of red wine in order to get enough resveratrol. So it's actually a purified component of red wine. But it does actually work, but it depends on genetic background. So those are the kinds of things that we're doing. So when I tell people about my research and we're trying to find drugs to make people live longer and things like that, usually the thing that they say to me is, that seems like a really bad idea. Why do they say that? Because they say, well, we already have enough people. And why do we want more old people? And not to be pejorative, but what they really mean is, why do we want somebody who's 90 to live to be 110? If we know enough 90-year-olds or 100-year-olds, their quality of life decreases late in life. And so that's a really valid thing to be concerned about. And so what we're really interested in is not addressing that problem. But this is an issue, regardless of our studies. Things are changing in the world and our society is not dealing with that change, I gotta say. So this is the predicted number of 65 to 84-year-olds in the red, and then 85 plus in the gray over time. And this paper was published, this figure, in 2014. So what you see is that the total number of people in these older age categories is going up very quickly. If you look at it as a fraction of the overall population, the prediction is by 2060, 25% of the population will be above 65 years old. Just as a touch point, when Social Security was created after around the World War II, only 1% of the population lived over 65 years old. So that is part of our economic challenges, is that the world is changing. We haven't really changed our societal expectations and norms. This is also a biological problem. So we don't necessarily just want to make people live longer. What we really want to do is make them live healthier longer, because that will address the healthcare needs as well as the quality of life of people. So what we would really like to do is imagine, instead of just living to be 100, that we would, right now our health declines rapidly after 60 years old. What we would like to do is push that health span out. So instead of you thinking about a 90-year-old as being a current 90-year-old, a 90-year-old would be more like a 60-year-old in terms of their health status. Then people say, oh, that seems like a good idea. And then we can continue to think about pushing that out. And again, in the questions, we can talk about still whether or not this is a good idea, but I think it's worth thinking about what our change in society will be as we do this, because this is going to happen one way or the other. I don't want to go too long. I think we're good. So we are looking at this as well in worms. And so there's, how do you tell if a worm is healthy? One way to do them is ask them if they can swim. And so if you do that in a 6-day-old worm, they swim really well. And then a 12-day-old worm is not decrepit, but older, and their swimming ability goes down dramatically. So one drug that was found in that mouse study that is now being used in humans, as a matter of fact, some humans are self-administering it, is a drug called metformin. Metformin is a diabetic drug. It's used by people with diabetes, so it's already FDA approved, so people can get a hold of it. And so when we treat worms with metformin, they do in fact still decline in their swimming ability, but that decline is less. So they are healthier longer late in life under metformin. So those are the kinds of drugs. And actually, we're not so interested in extending lifespan per se. What we really are looking for is ways to extend health span, because that will actually drive down medical costs as well as making people's lives better. So that's really our focus. So as I've done this work over the years, I decided that we were actually doing the wrong thing. I do think that, we'll address cancer and heart disease, and lots of people are doing great work in that. Maybe we'll make our bodies better. Maybe we can swim a little bit longer. But there is one problem that we have no way of addressing right now. And so I've decided in the last little bit of my life as a scientist that we're going to start focusing on this. And that is the brain. So here is a mouse brain. And this is not an animation of a brain. This is an actual brain that has been colored. Each little dot that you see is a different cell. It's a different neuron or other cell in the brain. And so what you see, and you know this already, I know, is that the brain is incredibly complex, but it's made up the same way that that frog embryo that I showed you. It is many, many different cells that are all connected to one another and that talk to each other. And the essential problem, again, this is a real inside this mouse's brain, tracing out the connections among all these cells. So the problem of aging and dementia is that these connections start falling apart. And the real problem is that we can't regenerate a brain because the cells don't regenerate very much. And more importantly, who we are as people is not just the fact that we're a collection of brain cells, but it's the pattern of connections between the cells. That is built on our own experience, our learning, whether or not we learn to play tennis or play a musical instrument. That's all in the connections of our brain. So even if we could replicate the cells in the brain, it still wouldn't be us. So this is really the problem of aging now, and it will be more and more. Why is it a problem? Because right now, 1 in 10 people over 65 has Alzheimer's disease, 32% of people over 85. Right now, the cost related to treating Alzheimer's disease is $300 billion. It's estimated that given the current projections and those changes in demography that I've seen, in current dollars by 2050, the annual cost to the US of dementia-related diseases will be $1.2 trillion. So we have to think about how we're going to address this. And this is really an issue, and it's not a scientific issue alone. It's a moral and societal issue. So how are we getting at this? I'm at the end of my time. We just got a brand new NIH grant that I'm very excited about. It's an exploratory grant. So these are more worms. It's a great Halloween thing. So the big worm in the middle is a female, and the little guys around the side are males. And so I don't know how many children are in the audience, but this is a thing called sex. So the worms are having sex. And one thing that we know about male worms is you don't have to ask them questions about what they're trying to do. This is what they want to do all the time throughout their life. And so we're starting to do a screen for mutations that allow old worms to have sex late in life. Well, that, again, great headline. Why are we doing that? It's because we want to find genes that help the neurons last longer late in life, because worm neurons, it turns out, are essentially the same as your neurons. The way that they work, the proteins that they have, all of those things are the same. So we're hoping to discover some proteins that help our neurons live longer, healthier, and then that might be a path towards, we're never going to cure dementias because eventually things are going to peter out, but maybe we can help extend our brain life, because at the end of the day, that's what we're really interested in as people. That's kind of how we define ourselves. So I'll just wrap up with this graph, or not graph, a picture from Dorothy Coleman. This is on her 105th birthday. And when asked the secret to her long life, she said simply, just keep smiling. So I do encourage you to just keep on smiling. At the end of the day, we can say, should we be doing this? Should we extend life or not? It will happen. We are living longer regardless. And so it's really a question of not whether or not we will live longer, but what will we do with the longer lives that we have? And that is not a scientific question alone. It's a moral question. It's a societal question. There are many aspects. And so the science is not going to answer the question of, how are we loved? How should we love? Those are other parts of our culture, like involving music and philosophy and those things. And that is another important part of what it is to be a University of Oregon, is to address those issues. But I will say that we are not prepared for these changes. They are happening to us now, and we need to engage and lean into that. And so I'm really appreciative of you being with me here this evening to hear about this and to start thinking about it. Because at the end of the day, all of these decisions are going to be made by you as citizens of the world, not by the scientists. So I'll wrap up with that, and I'm happy to answer any questions that you might have.
Great. In order to keep the questions a little organized, if you just want to pop your name right there into the chat, if you want to ask a question, and I'll just call your name out. And hopefully it's a lot of very interesting information there. So I'm assuming that we've got a few that would like to ask some questions of the provost.
I see Ellie has a question. Do you want to unmute and speak up?
I have two questions, actually. One, do the worms that you do research on, do they have the same set of telomeres as human chromosomes do? So that's my first question.
Yep, that's a good question. And so for those of you who are asking, first wondering, why such a technical question? What does that even mean? So the ends of our chromosomes, every time the cell divides, we replicate all of our DNA. And what happens is that there's a special protein that adds the ends onto the DNA. And so in our bodies, that protein actually gets turned off. It's probably an anti-cancer agent. And so slowly, every time our cells replicate, our chromosomes get shorter and shorter, and then they start eating into each other. So one approach to anti-aging, probably one component of it, is to think about extending those telomeres and protecting them. Unfortunately, a common feature of cancer is that telomerase, the protein that adds the ends, gets turned on, so it makes the cells immortal. And so there's kind of a trade-off there. Worms don't have exactly the same, they do have telomeres, but it's not exactly the same condition. One of the weird things about worms that is very different than humans in this case is that they have a finite number of cells and cell divisions, so that part of their biology is very different. A worm only has about 1,000 cells. And that's partially why they're studied, is we know every single cell. I can go in and look at every single neuron and know what it is. We cannot do that with you because all of us have different cells and the connections are all different.
Thank you. My second question is, I was reading an article just a couple of days ago about scientists in Russia unfreezing prehistoric roundworms.
Yeah.
And so they're claiming that they're probably around like 40,000 years old. So I was just wondering if you think that that's possible, or if you think it's more of a coincidence that the sample is contaminated, potentially.
Yeah, I just saw that headline. I haven't had a chance to. Great, these are like, that's a great Halloween story, and that's the actually the headline that I saw was on Twitter, and the person who quoted that article said, "Scientists, really in 2020, this is what you're doing, is unearthing 40,000-year-old worms that may come and eat us all." But I haven't looked into it. I do think it's conceivable. We freeze our worms all the time. They live a very long time in the freezer. So if you get them cold enough, they're small. And so the big problem with freezing, like us, if you tried to freeze us, is that our cells rupture. But these worms are small enough that you can actually freeze them without them rupturing. So it seems conceivable. We have been able to get DNA out of, for instance, even humans that are up to 40,000 years old. So some DNA stays intact over that time period. But all that being said, it's possible, but I'm still pretty skeptical, I gotta say. So,
we do
yeah, I was going to say we have another question about does diet and exercise affect longevity in worms and mice, and it does. It's a good thing to do. I will tell you two stories. The first is that you can make worms exercise, so you can force them to swim. And if you force exercise on them, they do live longer. It's not a hugely dramatic effect, but nothing compared to the genetic changes that we can induce in them, but it does work. And I think the same is true for mice. But there is one universal that makes every animal that has ever been studied live longer, and that's to reduce their calories, to reduce caloric intake. And so in particular, forcing animals close to starvation is very effective at making them live longer. And so, especially that's true in worms, and that's part of this whole pathway, presumably. It works really well in mice. So humans have tried it, in order to get the caloric restriction low enough to have the same effects that are observed in mice, people just decide it's just not worth it. They'd just rather die than eat that little food. So part of probably what we think we're going to find with looking for these drugs are mimetics that turn on the same pathways that caloric restriction does. And so that it's not the restriction of calories per se, but the induction of the stress response to starvation that's causing it. And probably some of you are even undergoing these periodic fasts and that kind of thing, which is now all the rage for losing weight. I simply can't do it myself. I just get lightheaded. But it's possible there's not enough information yet. It's possible that kind of cycle could lead to increases in longevity because of these same kind of caloric restriction effects. We don't know how long you have to do that. How far off something from market? So if I was making pitches, one of the hottest private equity industries right now is aging. So people are spending a lot of money, and there's a lot of hundreds of millions of dollars coming into the aging drug field. So people are starting to make bets on what could make a difference, because it's a pretty good roulette wheel if you think about it. Because if you hit on something, you're going to make a lot of money because everybody's going to start taking it. There are some, if you look on the internet, there are some, again, FDA-approved drugs that exist, that is, they're deemed safe for human consumption, but there's no FDA trial for aging. And this is actually a point of controversy in the field. The FDA will not actually okay a study for aging because they don't consider aging to be a disease. And so, so it's an interesting conundrum, catch-22 we're kind of in. That being said, there's a lot of work, and human aging biologists are really pushing on this. So metformin is one of the drugs, and there are people who are self-medicating on that. I don't recommend anyone self-medicate. I'm not a medical doctor, and we don't really know the ramifications of this. We could talk about, there's already evidence for some negative ones. There's another drug called rapamycin, which some people are using. And there's some other things that alter the energy metabolism in your cells that some people are also starting to take. So some of those are probably likely to work, and this is like early days, so we don't know. But because of that, kind of like the wild, wild west, there's going to be a lot of snake oil that's going to be sold in this space as well. And so it's really important that we take a scientific point of view.
Great.
Can I answer another question?
Yep, go for it.
So does genetics impact the likelihood of someone having Alzheimer's disease, and how would that impact my research? There is evidence that there is a genetic basis to Alzheimer's disease. One of the really frustrating things about Alzheimer's disease in particular is we don't even know what it is as a disease. So what we have are some markers that are associated, and you may have heard of these plaques and things like that, that are found and were discovered to be in the brains of Alzheimer's patients. The problem is we don't know about cause and effect yet. So we don't know if the plaques come after Alzheimer's disease or the cause of Alzheimer's disease. Matter of fact, there's mounting evidence that they're just a byproduct. What we do know is that there's a couple of changes in neurons that seem to be responsible. One helps maintain the integrity of the little tubes that maintain the connections, the dendrites between things. And when those start falling apart, then you start losing that. And the other is the thing that leads to the plaques, which are normal proteins that start sticking together, kind of like bad pasta, and they start sticking and sticking and sticking. And it's thought that that could be toxic to the neural cells as well. So we made a lot of progress on dementias, but again, we really don't even know why they happen. And if you look at my chart, dementia is really a very modern issue, although the people who now have dementia were probably developing it several decades ago. And that's also part of the challenge. We didn't even know to look for it, certainly 50 years ago, 30 years ago. So I think future scientists will have a lot more to work with. In terms of my own research, if we can find some genes that are protective of neurons, it might be interesting to see whether or not those are at a deficit in dementia patients. But the goal would be to see if we can find things that help the worm neurons. Can we then find some drug targets that will then turn on and upregulate those things in normal neurons? But you can well imagine that this is a decadal thing, because you'd have to test it in mice and then do clinical trials in humans and so on and so forth. So we're not going to deliver this next year for sure.
Okay, well, we're just about at time. Does anybody have one last question? You've stunned them all into silence.
We have one more in the chat, actually.
There we go.
From Kelly.
Yeah, so where do we get our worms? Yeah, so one of the great things, as I said about worms, is you can freeze them. So we can get them, and some of these have been collected over actually a 30 or 40 year period and then kept in people's freezers. So there are now people who have kind of all the collection of worms from all over the world in their freezer. The worms are very small. I didn't really talk about that. They're smaller than a grain of rice, so they're pretty easy to take care of and raise. They don't take up a lot of space. Some of the collections that I got from a colleague of mine in France, who on her vacation goes out and she always chooses an exotic locale and then collects rotting fruit. It's as a French scientist might do, I don't know. So then she brings that back into the lab, and more often than not, she discovers a new species of worm on there. She's a molecular biologist, but this is what she does for fun. And then because she's become famous at this, people just start sending her rotting stuff, and she looks through it and looks for worms. So that's more the classic model of the mad scientist, I think, that you might be thinking about.
So you don't have people just randomly sending you worms in the mail?
Not me. There are people who are now known for that, and so they are known and get random worms. But luckily, I am not. So no one's sending me just random rotting fruit from here and there.
Excellent. Okay, well, I think that puts us at time. Thank you so much for joining us this evening. We do have a little something for our provost here. Of course, we're all virtual, so I can't hand it to you, but we have a little something from one of our Portland duck-owned businesses, and it's en route to you, so you will have that shortly.
Thank you very much. That's totally unnecessary, but much appreciated. Yeah.
Well, we appreciate you spending your time with us. And I think, Ellie, am I going to hand it back over to you so we can award some gift cards?
Yes. So thank you again for everyone for attending. Huge thank you to all of our members as well. Awesome presentation tonight. So the winners of the $25 gift cards, so there's two of them, to the duck store, will be Kelly Cook and Chuck Norton. So if you can just send me a quick message with the email you'd like us to send it to, and then we will get that over to you. Thank you again for everybody for attending, and just follow us on social media and your emails for upcoming events. Go Ducks.
Great.
I was going to say, go Ducks.
Yes, go Ducks. Thank you all.
Yeah, you can just send me. Perfect. Okay, awesome. Thanks, Kelly. We'll send it out probably in the next day or so. And Chuck, what email would you like it sent to? Oh, sorry, you're on mute, Chuck. There you go.
Have it. It is CNX as Chuck Norton X-ray 2011 at G as in good